Okay, good morning. Hopefully, you can see my screen. And I'll introduce myself. I'm Waseem Qasim from Great Ormond Street Hospital and UCL in London. And Synthego have asked me to kick off by talking about applications of CRISPR for gene and cell therapy application. And I'm going to speak about uses of the technology for editing T cells in particular. And then in this session, we have Don Kohn from UCLA, who's going to talk about editing blood stem cells. And then we have a recorded presentation from Matt Porteus on editing in sickle. So I'll get on and present my slides, and then we'll do a Q&A and move on to Don's. So just in terms of introduction, I'm going to fill you in on CAR T cells. I'm aware it's going to be a mixed audience, so we'll give you a brief introduction to therapies using engineered T cells and how they've been reaching almost routine clinical applications now. And then I'll introduce the concept of editing T cells to try and improve the way they work for some of our applications in leukemia. And I'll then introduce CRISPR editing of T cells, where we had some help from Synthego along the way, and finish off by bringing up to date with applications involving base-edited CAR T cells and how we think they might be useful going forward. So look, by means of introduction, it's useful just to think for a minute why we are focusing on T cells and why so much of the field likes these cells for gene-type therapies. And there's a number of reasons listed on the left, which are all, they all begin with E. So they're easy to collect these cells and manipulate ex vivo. There's extensive literature and characterization over many years, both in mice and in humans, about how the cells work and how we can exploit them. There's extensive experience from transplantation, bone marrow transplantation in particular. And these are living cells that can exist for a long period of time. They can move, they can migrate, they can enter sites of tumor activity which antibodies can't reach, and they can provide long-lasting effects. And of course, T cells have some special properties. They exhibit what we call memory or recall effects, and they can also control and manage responses to their regulatory activity. And there's one more reason which I think is important to mention, especially for this particular field, is that you can do quite a lot to T cells. You can engineer their genome, you can break the DNA, they will tolerate translocations, but they won't easily transform. So there's very few examples of transforming mature T cells. And that's been a particular a plus for the field, that we've managed to engineer these cells without causing too much trouble. But of course, T cells, for anybody that's done even basic immunology, will know that T cells operate in a way that's only useful if you can avoid causing graft versus host disease, because T cell effects are quite tightly HLA restricted, meaning they are a very bespoke therapy in the conventional sense. So in terms of how we've been over the past decade or so trying to engineer T cells, on the left is an example of what a T cell receptor usually looks like, with a alpha-beta heterodimer recognizing HLA and presenting antigen on its surface, which has been processed within the cell and recognized in a very specific manner. And on the right, we've got engineered receptors, which are chimeras of binding domains from antibodies fused to activation domains. And the ones I'm going to really focus on today are second generation chimeric receptors, which have an activation domain derived from 4-1BB and CD3 zeta. And they've been the most useful for us so far. Now, you'll have seen slides like this before as well, that CAR therapies operate by recognizing malignant cells. This particular example is recognizing CD19 on the surface of a B-cell leukemia. And a single CAR T cell can go on to kill many multiples, dozens, maybe hundreds of tumor target cells, one after another. And the first licensed products have reached routine healthcare They're not cheap, they're expensive, and they're bespoke, so they're manufactured for each patient using their own cells. But some of the response rates for certain types of leukemia and lymphoma have actually been quite impressive. And they've changed the face of how we deal with some of the harder to treat leukemias. But as with all therapies as they emerge, there are various limitations. And this useful review from last year summarizes some of those. There's side effects. There's a difficulty in addressing antigen loss if the target disappears off the surface of cells. And there's this issue about having to make bespoke products for each individual patient and the risk of those products not working because they're from cells that are not perhaps the healthiest, and the issue of being able to deploy this to large numbers of patients across many different hospital type settings. And so we can try and address some of these by introducing genome editing steps. And the first examples of those are now being published in the literature. We described, and I mentioned on the next slide, the application of TALEN reagents to make what we call universal T cells that can be used without matching. There's similar examples emerging using other homing endonucleases, and the first trials are being reported. And then at the end of last year, the first reports of CRISPR-engineered T cells from Carl June and others with recombinant T cell receptors, and reports of PD-1-engineered cells in the use of not CAR T cells, but tumor-specific T cells in lung cancers were being reported. So the technology has rapidly gone from discovery to being applied in the first clinical trials. So the kind of effects that you can elicit with engineering range from addressing the issue of expressing a T cell receptor and wanting to remove that. We can remove HLA molecules and other molecules that are targets for some of the drugs that we use during conditioning. This one's of particular importance to us, CD52, target of one of the antibodies that we use. Or you can target PD-1, which is part of the checkpoint pathways, and promote persistence. And then there's the perennial question of how you introduce these into cells, whether you use viral vectors or whether you combine them somehow with electroporation steps using either mRNA or RNP complexes. But in any case, the first examples are now treating a couple of dozen patients or so using TALEN reagents. And they've shown us the way in terms of how we can prepare the patient and the cells to get quite powerful anti-leukemic effects in both children and in adults. Excuse me. So our ambition has been to switch from TALEN to CRISPR and take these things into trial as a proof of concept that the technology is useful. And when we started this particular application, in fact, I'm not sure Synthego had started providing guides at that time, and there was an issue about how we would deliver guide at high efficiency. So what we did in the first instance was to incorporate it into the long-terminal repeat region of one of our viral vectors that was also expressing a particular CAR transgene. And that has the added effect of allowing us both to knock out and transduce cells at the same time. The Cas, by the way, in this instance is delivered transiently by electroporation as mRNA. And that particular system has been advanced all the way to GMP manufacturing and is ready for clinic. We use this device, which allows us to engineer hundreds of millions, if not billions, of cells in one batch. It's called a prodigy device, and it allows us to culture and expand cells and select them if we need to. And then electroporation-wise, there's this device from Lonza that's been upgraded to work in a compliant manner. And on the right here, I can show you some flow plots of the type of cells we end up with at the end of a weeks-long manufacturing process in one of these devices. So on the horizontal, for those of you who are familiar with these kind of flow plots, we've got this staining for CAR expression, which is, in this example, is around 90%. And we've got very low levels of residual expression of the two targets that we've knocked out, which are the T cell receptor and CD52, virtually no expression at all. And so we've taken this particular platform forward to the first trial applications. And to do that, we've been screening and looking in some detail to see what off-target effects might be occurring or what undesirable effects might be occurring. One thing we can do is look for predicted effects when we're knocking out multiple genes. So this is a translocation PCR. And we can see at low frequency these events do occur, but in some they're probably less than 1% of the overall population. And that's well within the range that we saw with previous TALEN editing. And we can do more sophisticated analysis at predicted off-target sites and look at high-depth analysis, looking for off-target effects. And we've done that both for our TCR and CD52 sites. And for these particular targets, find only very low levels of editing effects. And that's been quite reassuring as well. And so in the UK, in London, in fact, we've just opened a study to test these cells in the same setting as previously with the TALEN cells. These are children with relapsed B-cell malignancies, and they'll receive some conditioning regimen to prepare them and lymphodeplete them. A single dose of CAR T cells is proposed, and then we'll be tracking them to see how their disease responds or clears. And the ambition is to get them into remission and allow them to have clearance of disease without side effects such as cytokine storm that we sometimes encounter. So that's CRISPR, and it's come very quickly from laboratory through translation into phase 1 in a very specific context where we think, in fact, we can test its safety and inform further studies. But at the same time, the technology has moved on, and you'll be aware that there are variations of these technologies that can now edit the genome without actually causing the double-stranded breaks that we see with CRISPR-Cas9. So on the left here, and you've heard already this morning about how these enzymes work, we can either end up with a knockout or we can deliver a homology repair template. But from MIT 2017 were these descriptions of chimeric proteins comprising deaminase enzymes linked to a deactivated Cas and a UGI element, which now allows us to very specifically cause C to T changes, and we can exploit this to, at quite high efficiency, introduce either premature stop codons or disrupt splice sites to knock out genes in the same context as creating the non-homologous changes there, but without the double-stranded break. And in fact, it's been relatively easy to adapt this and take it along the same route as we have for CRISPR. Here's some examples where we can disrupt the T-cell receptor expression, we can disrupt that target I mentioned, CD52, or we can just disrupt class 1 expression, all at relatively high efficiency, considering we're dealing with large numbers of cells in a scale-up manipulation system. So how might we want to apply this? Well, I think one of our first applications is going to be for multiplex editing of T cells. Here's one example where we're trying to engineer groups of cells that will work cooperatively without attacking each other. So we need to remove multiple antigens at the same time. And the example here is showing removal of an antigen called CD7, removal of the T cell receptor, and we'd also remove CD52. And we can do that at high efficiency without causing that problem we saw with the previous issue, with the previous iteration of Cas9, those translocation events. So we can see here, if there's 4 possible translocation events at 2 cut sites, with the Cas, as I showed you on the earlier slide, we see up to 1% translocation events, but with the edited, base-edited T cells, we see virtually undetectable levels of conversion going on there. So we think, in fact, that particular platform will be useful going forward for our next iteration of engineered cells, which will be used to in the UK and Europe, in Germany, UK, Germany, will be used to treat children with relapsed AML, where we need to have multiple edits in cells. It's a particular problem, this, because in our patient cohorts, we have pretty dismal survival. You can see from these curves here that the survival rates are very low in these patients. And so as a good proof of concept application, that is the one that we're advancing to trial next in that same context of relapse ahead of transplantation. So I hope I've given you a flavor of the kind of applications we can expect from CRISPR in the first instance, and our particular interest is T cells, which are ideally suited to being handled and manipulated ex vivo without too much worry about causing side effects. And we can move quite quickly from conventional CRISPR to other forms of editing using the same type of guide technology, especially for multiplexed effects. So I'm trying to stick to time. I think that's just about right. I'm going to acknowledge members of my group and funding bodies that have helped us along the way. And then I'm going to stop sharing the screen and see if there's any questions on the other screen. So I've got a couple of questions here, which I think are very reasonable. So first one here from Justin, who is asking, what about doing this with MHC? And the answer is yes, we are doing this with MHC. There's two sets of MHC molecules, obviously. There's MHC class 1 and there's MHC class 2. And we have got ways of targeting them so we get disruption of both molecules, and we can do that at high efficiency. I think those will come into play for some of the forthcoming studies. They're not in the current versions, but they are in the pipeline. And then expanding on that slightly, there's plenty of other groups, including ours, that are looking at taking MHC off for regenerative medicine applications from iPS and other types of stem cells. So that's definitely in the pipeline. Okay, there's some more questions here. How are you delivering base editor to your T cells? Well, in fact, as I alluded to, the technology for delivering both the Cas9 and the base editor, if it's as mRNA, is by an electroporation step at the same time as the delivery of guides. And that's, we found, has been the most efficient way. And of course, if it's electroporation and it's mRNA, it's a temporary delivery, and the effects are mediated within about 24 hours, 48 hours tops. Okay. Do we have time to take any more? Should I move on? I think I should move on to Don to keep it to schedule.
Screen. Okay, I'll just take it from here. So hello, thank you, and I'd like to thank Kevin and other people from Synthego for inviting me to talk. I'm going to try and cover two kind of major areas. The first half will be just talking about work going on in my lab over the last 5-6 years developing editing in hematopoietic stem cells. And the second half will be work that we've done collaboratively with investigators at UC Berkeley and UCSF, Oakland Children's, developing a clinical trial of editing for sickle cell disease. And as Jennifer Doudna introduced in the beginning, sickle cell is a very attractive target for editing because every patient in the world with the disease has the same single base pair change. And so it really is, a single approach would be very applicable across all patients. And so the mutation is in the 6 codon, that number code for a negatively charged glutamic acid. There's a T that makes a code for a hydrophobic valine that causes the hemoglobins to aggregate. So all we need to do is add a donor that's complementary, but it has a corrected change to revert that base. So we started this work again about 6-7 years ago, working with Sangamo with zinc finger nucleases initially, and we developed a system in the laboratory to start with hematopoietic stem cells, do the editing event, in this case with ZFNs, now more recently CRISPR, and adding the donor of some type, and then doing an in vitro differentiation to make red cells. Then we can look at expression of hemoglobins. And so if we do this with bone marrow from a patient with sickle cell disease, we see that untreated, we see they only make hemoglobin S and maybe a little bit of F. Now my pop-ups aren't working either, I guess. Okay, well, what you're not seeing is the peak that after we've edited them, there's a peak of hemoglobin A that appears. And so the good news was that we could make some hemoglobin A, but in fact, when we looked at the edits that occurred, while we had about 15-20% of corrections of the mutation, this is you're using an IDLV, an integrase effective lentiviral vector, to deliver the donor sequences, or a single-stranded oligonucleotide. We had fairly high rates of indels at the target site. So we had a relatively low ratio of correction to disruptive non-homologous end joining, which is obviously not interested. in the middle of a coding region like for beta globin. So that sort of started us on a quest to find a way to increase homology-directed repair. And so over the years, we've tried many different things to make this work. We Matt Porteus, Paula Cannon, and other people reporting that AAV6 as a donor gave high rates of HDR compared to what we had been using with an SSODN. So I'll show you a little data on that. We've tried overexpressing a variety of the limiting factors for homology-directed repair by transiently transfecting the messenger RNA for a variety of proteins, none of which worked even in multiple combinations. We've also made multiple fusion proteins to Cas9 to try and bring in HDR factors, and really none of those have worked other than the box that was displaced. Geminin fusion that I'll show you has actually improved the editing outcomes. We've done and are continuing to do multiple efforts to sort of arrest the cells in the favorable stage of cell cycle for HDR to occur, SG2 or G2M. And for the most part, we found that hematopoietic stem cells don't like to be cell cycle arrested. And I'll show you a little bit of work with that actually media does make a difference. And so for the work that I'll show you, this is the sickle mutation site. So we use donors where the guide binds over here. We have a mutation in the donor and the PAM site so that we don't recut. We either looked at delivering this by a single-stranded oligonucleotide or as an AAV6 carrying this 1.1 kb fragment from the 5' end of the globin gene. And so when this is, let me walk you through this slide. This is where we took CD34 cells, we edited them using either an oligonucleotide or an AAV6 as the donor, and then transplanted them into immune deficient mice. And so when we look at the cells edited in vitro before we transplant them, the white bars are HDR. And so there's much more HDR with the two different AAV donors than with the oligo. And this is just the ratio of HDR to NEGJ. So the oligo is much lower. When we transplant the cells into mice, we actually consistently see lower engraftment with the cells exposed to AAV than the cells exposed to the oligo. The differentiation of the different lineages is unchanged, but in fact, in vivo, the editing HDR ratios were not different, despite the big differences we saw in vitro. And so we think this means that the biology of the engrafting stem cells, HDR is limited, and it doesn't matter how much donor you've got in there. And so our conclusions were AAV achieved higher HDR in vitro, but AAV reduced engraftment in our hands, and it may be the preps of AAV that we're using. HDR in vivo was reduced from in vitro, and the AAV and the oligo were not different. And in this work, Dr. Romero also did RNA-seq on the cells edited by these different methods. And so you can see that compared to mock-treated cells, the oligonucleotide did not change the expression patterns, principal component analysis, whereas the AAV6 delivered donors did have a bigger change. The same thing when combined with RNP. So the oligo had a modest change, whereas the AAV6 combined with the RNP had a bigger change. And you can see that here, the oligo had a small number of genes change, and there were much more with AAV6. And some of these may be involved in what we saw, the limited engraftment with AAV6. And so focusing more on the cell cycle, it's relatively known that throughout most of G1 and into S phase, the DNA is dispersed, but then it becomes condensed prior to mitosis. NEGJ occurs throughout the cell cycle, whereas HDR is confined sort of the late S and G2 phases. And so, in fact, when we've looked at editing by cell cycle, we see this difference. So here we electroporated CD34 cells with CRISPR-Cas9 and an AAV6 donor, and then the cells were immediately sorted by their stage of cell cycle they were in at that time, and then they were grown out, and then the genome was sequenced. And so we said the cells that were in SG2 at the time of editing had a much higher rate of HDR than the cells that were in G1, resulting in a much improved ratio of HDR to NEGJ. And so one option would be just to sort those cells out and use those. That would be a subset of the cells that might not be enough for a graft. And so we've then looked at what I'd mentioned earlier, this fusion protein between Cas9 and geminin. And if the slide will open, So geminin is a protein that's destabilized in G1 phase of cell cycle by ubiquitination. And when you put a fragment of geminin onto a protein, it will make it unstable through G1. And as I'll show you, that actually then, because it limits the activity of the nuclease to the SG2, it decreases the amount of NHEJ that occurs. This was work that was done by Anastasia Lebova, who was a graduate student at the time. And so you can see that with in the hatched bars are the geminin Cas9. HDR was not different, but there was a significantly lower amount of NHEJ, again, resulting in an improved ratio of NHEJ to HDR. And then finally, we've been looking at media and what effects they have. This was in part spurred by a paper from Saksida Ravan and Harry Malik, where they had very good editing outcomes, and we looked at their media comparison. So we compared our standard medium conditions to those that they reported in a paper where they had very good editing rates with an oligo. We call their conditions the full Malik. Then we looked sort of halfway between, or the half Malik. And so our conditions had been ex vivo 15 and various cytokines at 50 nanograms per mil, whereas they use SFEM2, slightly higher amounts of cytokines plus IL-6 and other small molecule additives. And so we edited cells by our standard method with these conditions. And then this is the final readout of the cells transplanted in immune deficient mice. So you can see in blue, the cells edited under the Malik media conditions had about 5-fold higher rates of HDR and a smaller fraction of cells that did not have editing in them. So the media does plays a significant role in what types of edits that you get. And Dr. Romero is continuing to follow this and has significantly more work showing the improved outcomes just by changing to the medium of SFEM2. So for the remainder of the talk, I want to talk about work that we've been involved with in a really great collaboration with Mark Walters, David Martin from UCSF, Oakland Children's, Jacob Kohn, who was at UC Berkeley, Mark DeWitt, who's really been doing the heavy lifting for the project, Wendy Magis, who's one of the scientists who did a lot of the basic cell biology, Salima Romero, who did the translation, and Fyodor Urnov, who's leading the work of the IGI for the sequence analysis. This has been funded by CERN in 2 successive stages. And the goal of this was to develop a clinical trial to use the CRISPR editing, as I've shown you, for sickle cell disease. This was based on work published by the group up in Oakland and Berkeley, editing using Cas9 RNP and a single-stranded oligonucleotide donor to edit the cells. And then so the clinical schema would involve making a patient's cell product by editing, and then once you've released the cells as being conforming, conditioning the patient and infusing the cells. And so again, it's the business end of the donor. This is where the guide binds. It has a PAM mutation to prevent recutting. Then it has actually 2 base pairs that will change it to a glutamic acid, which is the wild type codon. And so when this editing is done just at small scale in CD34 cells for patients with sickle cell, again, the unedited shows only HBS, but in 2 separate patient samples edited by this method, the amount of S is way reduced and the amount of A comes up. And so this manufacturing then has been transferred from the laboratory into the GMP. And Salima Romero and her group, again, led this tech transfer. So now we're editing between 0.1 to 1 billion cells per lot. And we're using the Clinimax to enrich the CD34 cells and the MaxCyte electroporator for our electroporation. And then the drug substance, which are the edited CD34s, is cryopreserved to make the drug product, which is release tested and then can be infused or thawed for infusion. And in the work up to developing the IND, we edited lots from 3 sickle donors. I showed you 2 examples of that, and 3 large scale, at a clinical scale amount of cells from healthy donors. And these are the editing outcomes. So we had good viability, averaging 81%. The cells are healthy in terms of the progenitors being able to form colony-forming units. Our correction of the sickle mutation is approximately 26%, either in the sickle patients or just reverting in the healthy donor. And the indels were 50%. So very consistent patient-to-patient levels of editing that we're really pleased with. And then we've looked at the alleles in the product. And so we see in blue our corrected alleles, red our indel alleles. And so you can see that over half the alleles in general are corrected, and about 40% or so have indels in them. And then in terms of the specificity of this guide, so looking by guide-seq in multiple experiments in 2 different cell lines, we see that there's only one recurrent off-target site. It's sort of an intergenic region with nothing of concern in the region, but we do get about 3% off-target cutting at that one site in 2 different large-scale runs. Using bioinformatics, 190 other putative off-target sites were predicted and analyzed by sequencing and targeted resequenced those sites. And really there was no off-target activity in any of those. So just the one off-target site was seen. And then the cells were, from some of the lots, were engrafted into immune deficient mice. And again, we saw very good engraftment, both unedited or the edited cells. The amount of HDR was about, again, what we'd seen before, averaging 22.5%, and the indels averaged 58.3%. So we're seeing, again, very consistent editing activity in the patient lots. We've also done a large-scale toxicology study where 50 mice per group were engrafted with either edited or non-edited cells. And there was really just a slight decrease in engraftment with the edited cells. And the editing was as we had seen before, but there were no adverse events in terms of overgrowth of the cells from the editing. And so based on this, we've now developed a clinical protocol, transplantation of CRISPR-Cas9 corrected hematopoietic stem cells. We're calling the product CRISPR-SCD-001 in patients with sickle cell disease. And the goal then is to open a single dose, open phase 1 study in patients with severe sickle cell disease. We plan initially on 9 subjects, and Mark DeWitt, shown here smiling, just submitted our IND last week, and it was a lot of work for him, very heroic effort. So we're looking to the first patients in 2021, and then after the first 3 patients are treated, there will be a DSMB review, and if that is favorable, we would then lower the age of enrollment from 18 years and older to down to, I believe, 13. So that would be the expanded pediatric cohort. And so I'll stop there just to acknowledge all the people that have done the work. I showed you sort of the lead investigators at, again, this was sort of a multi-UC collaboration. These are the home institutions for all of us. And of course, none of this work can be done without the funding from multiple places, most prominently CIRM. A lot of our preclinical work was funded by the Doris Duke Foundation. We've also had support from the Cure Sickle initiative out of NHLBI. And I'll stop there. Thank you.
Don, that's a brilliant update. Thanks very much. I think I can see the questions. Can you see the questions? Yeah.
Yes.
Okay. Let's ask you a couple. How long? Ian Frenens asks, how long is the donor DNA template? I mean, I think you're using oligos, but do you want to answer the question more broadly?
So for the clinical oligos, 160, I think 162 base pairs in length. So it's sort of an it's a relatively long single-stranded oligo. Our preclinical work in my lab, we used one that was about 100 base pairs, and we see relatively similar rates of editing.
Okay, that's good. And then Jason asks, is there a risk about sickle cell transformation? could gene editing lead to transformation, I think is the question.
Right. Well, I think the concern for transformation with using CRISPRs and cutting that you would also hit on is the concern mainly about, either off-target cutting in a like a tumor suppressor gene or a translocation that could be transforming. And so I think it's going to be very guide specific. It's necessary for each guide that's used to some extent to do the safety studies to characterize its profile of off-target activity and make sure that it doesn't have, have off-target activity near genes that cause a problem. I don't know that sickle poses any more risk than other patients' CD30 hematopoietic stem cells would.
Yeah, okay, I think so. That's probably right. Okay, I think we've got one more. Dylan asks, I'm not sure if it's a he or a she, but asks about neonatal AAV editing of hepatocytes. And do you think that gemin fusion will work in other cell types such as hepatocytes?
I mean, yes, it should. I think the cell cycle machinery that regulates its ubiquitination is relatively universal, and so it could, have the same effect to decrease unwanted NHEJ in the hepatocytes. We haven't looked at that. It would be very interesting to look at that. And contact us if you want to. We can share our gene with you.
Okay, so you've got that message. Hopefully, to get in touch directly if you want to have that information. John, thanks very much for that. It's a very useful summary. Thanks very much.
Thank you very much.
Okay, now there's one more talk in this session, which is from Matt.
Hello, and thank you for joining us on World CRISPR Day. And I'm excited to be part of the second session talking about the tremendous impact CRISPR has had on translational genome editing. And congratulations to Jennifer and Emmanuel for their well-deserved award, Nobel Prize from 2 weeks ago. So I'm going to talk about our efforts in terms of using genome editing as a way of providing long-term, durable, and we believe definitive cures for genetic diseases, including sickle cell disease. I'm fortunate enough that there's a lot of interest in this field and involved in several different companies. The program I'm talking about today in terms of gene correction for sickle cell disease, we hope will be licensed to Graphite Bio for further development. So, we're really at a dawn, I think, of a new era in medicines. Sanitation, antisepsis, vaccines, small molecules, biologics have all transformed the lives of patients all over the world. But they have certain limitations, and there are still many diseases and many people who, for whom these modes of therapy are still not adequate. But I think we're at the dawn of at least two new types of living drugs, microbiome manipulation, which I won't speak about today, but the addition of using genetically engineered cells to give to people as medicines. And the reason I think these will really transform the lives of patients is that they're living drugs. They migrate, divide, respond to their environment in an entirely different way than these other modalities. Moreover, if you use a genetically modified or genetically engineered stem cell, it has the potential to give lifetime durability, which again is in stark contrast to many of these other modes. So the way we like to genetically engineer stem cells is to use genome editing. And the fundamental process is to use an engineered nuclease. And as we all know, the CRISPR-Cas9 system has democratized and really accelerated the ability for us to use genome editing because it is so simple to design a highly active and highly specific nuclease that makes a break at a specific site in the genome. Once the break is made, the cell then repairs the break in two fundamental ways, although there are subvariants of each of these ways, including by non-homologous end joining, in which case small insertions and deletions could be made at the site of the break, which allows you to specifically inactivate genes or genetic elements. And it's both being applied in a translational setting, but also has been a tremendously powerful research tool. But the pathway that we really are interested in harnessing is the homologous recombination pathway, in which the break gets repaired by the recombination machinery using a provided donor template. And the recombination machinery will use this donor template as a template or a substrate to make a copy and thereby paste the sequences in the donor into the site of the break. And in this way, one can create single nucleotide changes or insert large cassettes of genes. And so today I'll talk specifically about how we're using the recombination pathway to create small nucleotide changes as a treatment for sickle cell disease. But the process of recombinational genome editing can be applied in a number of different ways. One of the things that also is intriguing about genome editing is, before I go into the specific details of sickle cell disease, is there may be less failures, or at least early on, there may be less failures. As we all know, drug development is a highly risky, highly expensive process with reportedly only 10% of programs actually reaching the clinic. And part of the reason for that is that target identification and measuring the effectiveness of your drug at hitting the target are quite difficult. Whereas in genome editing, target identification, particularly for the genetic diseases, is done and well validated. And moreover, what I'll show you is that it is quite easy now to measure the effectiveness and specificity of our genome editing process at our drug target, which is, of course, the genetic sequence that causes the disease. So we'll see if this prediction turns out to be true. Another point is that genome editing, while we're adopting it for translational purposes and research, is actually one of nature's major ways of creating genetic diversity. And I've listed here several different natural genome editing systems that all proceed through the induction of a double-stranded break at a specific target locus. Perhaps the most notable of these is the way in which animals create diversity in their immune systems through VDJ recombination. And the irony, of course, is now the CRISPR system, which was identified in bacteria as creating an adaptive immune response to phages, is now mimicking what the animal immune system does with VDJ recombination. So the system that we have developed and have used is an ex vivo gene targeting system in which we purify our cell of interest. And we put it into cycle because the recombination machinery is most active in S and G2 of the cell cycle. We then deliver the CRISPR nuclease as a ribonucleoprotein complex. We use a high fidelity Cas9 protein, and I've listed two sources for this protein. And we complex this Cas9 protein to what I call an S-cubed guide RNA, SQ for synthetic, stabilized, and single guide RNA. And again, these guide RNAs now can be purchased from a number of different sources. We complex the guide RNA to the protein and deliver this complex to cells via electroporation. Now, to provide the donor template, we use a non-integrating adeno-associated virus type serotype 6. And this virus has evolved ways of delivering its nucleic acid cargo to the nucleus of cells without being detected. And this system is highly active, and I list a number of different publications from our lab demonstrating its activity and including a protocol that we still generally follow. And here we routinely obtain between 40 and 80% targeted integration in a wide variety of primary human cell types using this system. The only cell type where it appears to be a bit lower is neuronal stem cells, which are much more slowly dividing. So what we would like to do is apply this system in a number of different ways. And one could think about applying genome editing to add a new therapeutic function to a stem cell or a cell to give it, as I said, a new therapeutic function. And this would be a form of synthetic biology. But what I want to talk about for the remaining time is the idea of rescuing a missing function in a cell by modifying a variant that causes disease. And the specific disease I want to focus on is sickle cell disease. So sickle cell disease is probably the world's most common severe genetic disease. Every patient has the same mutation in codon 6 of the beta globin gene. A adenine is switched to a thymidine. This leads to a single amino acid change, a charged amino acid converted to a polar amino acid. polar amino acid creates a hydrophobic surface that in the deoxygenated state allows the near crystalline concentrations of the hemoglobin tetramer to polymerize, converting a round red blood cell into this stiff sickle-shaped red blood cell that gives the disease its name. And these stiff sickle red blood cells block the ability of red blood cells, which to deliver effective amounts of oxygen to the distal tissues, and leads to a multisystemic disease. In Africa, where over close to 300,000 people per year are born, the median lifespan is in the first decade of life, whereas in the United States and Europe, the median lifespan is in the mid-40s, still too short. So the disease is caused by red blood cells, but red blood cells have a very short lifespan, even in the non-sickling state. And so if one were to develop a long-term effective therapy for sickle cell disease, one would need to modify the hematopoietic stem cell, which gives rise to red blood cells for the duration of our lifetimes. And one way we give so-called corrected hematopoietic stem cells is through allogeneic hematopoietic stem cell transplantation. And in this, what we do here is we find a donor who's an immune match. We give the patient high doses of chemotherapy, and then we give the stem cells from the donor to the patient. And this was first done in 1984 at St. Jude's Children's Hospital, where a young girl with AML and sickle cell disease was treated with a bone marrow transplant from her brother with sickle cell trait. And she adopted the hematologic parameters of her sickle cell trait brother. My understanding is she's alive and well, both cured of her AML and cured of her sickle cell disease. And this has been broadly applied now and has been used over, and now a paper that's 4 years old described the success of using matched sibling donor transplants as a cure for sickle cell disease with a overall hematologic cure rate of 95%. The problem is that most patients don't have a matched sibling donor, and there's still major immunologic complications, including graft versus host disease and poor immune function. So what we'd like to do then is replace allogeneic cells, which prove that genetically corrected stem cells can cure this disease, and replace them with the patient's own stem cells that we directly correct using genome editing. And a large team of people, some of whom I show here, Annalisa, Rasmus, Danny, Job, and many others, have demonstrated that we can, in CD34 cells, which is a marker of the hematopoietic stem and progenitor cell, we can get gene correction frequencies of 60 to 70%, allele correction frequencies of 60 to 70%. Now, notably, in addition to creating correction, we create indels. And what ends up happening is you create a mixed population of cells in which some alleles are unmodified, some cells have both alleles unmodified, some cells have both alleles modified by an indel, some cells have both alleles modified in the way we want. And so you create this heterogeneous genotypic mixture, but we note that over 50% of the cells have at least one allele corrected, which is equivalent to sickle cell trait. And so that we think that that's a level that will be sufficient. And when we take the CD34 cells from a sickle cell disease patient and convert them into red blood cells, what we find is we essentially convert all hemoglobin S production, which is the pathologic hemoglobin, to over 90% hemoglobin A production. When we ask whether we've truly modified a long-term hematopoietic stem cell, we've looked at this in two different ways. One is by looking at the cell surface markers that identify true long-term hematopoietic stem cells. And while we find that the overall gene correction frequency is close to 80% in the CD34 positive progenitor population, in the phenotypic long-term HSC, it is 60% or above. So a small decrease, presumably due to HSC cycling a little more slowly than the progenitor cells, but nonetheless, a still very high number. So the other way we want to assess whether we've modified a stem cell is using an animal model. And George Box, who on his Wikipedia page is described as one of the greatest statistical minds of the 20th century, wrote, essentially all models are wrong, but some are useful. Or he rephrased that as, remember that all models are wrong. The practical question is, how wrong do they have to be to not be useful? And for me, our best current model is the immunodeficient mouse, NSG mouse model, as a way of analyzing the successful engraftment of human hematopoietic stem cells. Still not sure is whether this model is going to be useful or not in terms of predicting what will happen in human patients. And what we find is, and the reason I have this skepticism is, we find that depending on the CD34 cell type we use, we get different levels of engraftment following transplantation into NSG mice. So if we use CD34 cells from the circulation of sickle cell disease patients, we see essentially no drop in the engraftment of gene-corrected cells. Whereas when we use mobilized CD34 cells from healthy donor patients, we get in vitro our high levels of correction of 50 to 60%, but following transplantation, we get a median of about 20%, with incredibly high variability, with some mice having 70% corrected cells and other mice having 5% corrected cells. We take the same type of cells from sickle cell disease patients as generously provided by John Tisdale, and we find the median correction frequency is 30%. So there's a high variability in terms of just the engraftment into NSG mice. I've put here two bars. The red dotted line is what I call the Tisdale-Fitzhugh line, which suggests that we need over 15% alleles corrected in order to cure the disease. And I put here the Walters line, who, again, both of these groups from human patients, Mark Walters has demonstrated that patients even with as low as 2 to 5% correction can have a significant benefit, and maybe and hematologic parameters that are almost normal. So we've scaled up this process in order to begin a clinical trial, and I'm not going to review in detail this entire table, but through time, we've gradually scaled up the process so we can treat somewhere between 150 million and 700 million cells. The overall correction frequency has fallen slightly from that 60 to 80% range to more in the 40 to 60% range. But again, it's even exceeding the Tisdale-Fitzhugh line. And the products are highly, the specificity is quite high. In fact, I would argue that the specificity of our genome editing process might be higher, certainly higher than most, if not all, small molecule drugs, and higher than what are the natural genome stability of these cells. And we find that there's a single off-target site that is modified by our system, is modified at a frequency that is 1 to 2% of the on-target site, and it is located in a site of the genome of no known functional consequence. So what we have done is submitted an IND to the FDA to seek approval to move forward with a clinical trial in which we would harvest hematopoietic stem cells from a patient, purify them, and then manufacture them under GMP conditions, have that product undergo quality control, and if it passes our 20% allele correction and a minimal cell number, we would transplant those patients as part of an autologous transplant back into the patient after the patient received full dose myeloablative busulfan conditioning. I show a picture of a child here because one day we would like to use this in children before the end organ damage of sickle cell disease occurs, but initially we will start in young adults for ethical reasons. One of the things that people have worried about is whether we would have enough AAV6 to treat a large number of patients. And because we are in an ex vivo setting, and because the production of AAV6 is getting better and better, we actually now have a lot of AAV6 that would allow us to treat 10 adult brontosauruses, which translates into several thousand human beings. So the supply chain around the reagents, both the AAV6 and the guide RNA, is robust enough that single lots are now going to allow us to treat thousands of patients. With that, I want to thank you again for participating in World CRISPR Day 2020, a really exciting year to be able to present in this conference. So thank you for your attention and the opportunity to present. I want to thank all the talented graduate students, undergraduates, postdocs, senior scientists in my lab who've supported this work. The funding from a wide range of sources, including the NIH, California Institute of Regenerative Medicine, the Doris Duke Foundation, and a number of private entities. My fantastic collaborators in the Division of Pediatric Stem Cell Transplantation and Regenerative Medicine, our collaborators in the Stanford GMP facility who have taken what we've done in the lab and moved it into a clinically compatible process. And then finally, collaborators at Stanford and academic collaborators at Stanford and other universities, and collaborators in a wide variety of companies. It's been really rewarding to work with this a tremendous group of people. So thank you very much and enjoy the rest of the day.